to Rosa Chefaoui. Rosa did her PhD here, part-time at the museum. She worked on modeling the distribution of invertebrates, and she finished in 2010. Jorge Lobo and myself. And after that, she made a small career shift, and she got some postdocs. She loved the sea. So she got some postdocs and went to the CSIMAR in Faro, University of Faro in Portugal, for 8 years to work on applying species distribution models and many other modeling applications to sea creatures, and as you will see, to algae. Since 2021, she's a Marie Sklodowska-Curie Fellow at Juan Carlos, working with MARHOT, which tries to forecast the effects of climate change on marine macrophytes. And that's what she's going to talk about today. Thanks, Rosa.
Thank you very much, Joaquin, and thank you also to Rod Wilson for inviting me. And thank you, everybody, for coming. I will show here some of my research related to climate change and their effects on macrophytes, marine macrophytes. I will show you some examples of my research linked to these issues. And as you know, there's no breaking news that climate change is affecting to terrestrial ecosystems, but also marine ecosystems have been affected a lot. There's an increasing trend of sea surface temperature globally. This evidence comes from direct measures and also satellite data. You can see how the last Copernicus report also confirms that fact. So there are many changes in marine ecosystems. One of them is the change in biomass. There's an increased loss in biomass that, as you can see in the predictions, can only be stopped if we achieve the RCP 2.6 measures. Otherwise, it will continue increasing the loss of biomass. In this talk, I will talk about species that are foundation species. This means that they are engineered species which create habitats for other species, for example, acting as nursery for fishes. And this entails species such as seagrasses, coral reefs, mangroves, macroalgae. Most of them are responsible for the main primary production in the oceans. And they also supply many ecosystem functions in services for humans. You can see that there's evidence of the effects of climate change in these species. For example, this is Zostera noltei, a seagrass, which suffer a decrease in shoot density and change in color due to the change in photosynthetic pigments when a +4 degrees was applied in mesocosm experiments. There are also many papers documenting changes in distributional shifts of these species. Sea surface temperature follows a very marked gradient in the sea in comparison with land. This is the oscillations that are suffered monthly by sea surface temperature. And this marked gradient in sea surface temperature affects how these species can adapt. For example, you can see that when comparing marine and terrestrial ectotherms, it has been found that marine ectotherms, their niches conform better with their distributional ranges. Also, for a marine species, it is more difficult to migrate to find a suitable thermocline when a change in 1 degree is applied. in their medium because the isotherms are longer apart than in terrestrial ecosystems. So these facts make us to provide some confidence when we apply species distribution models to predict the shifts in distribution that species can suffer because of climate change. Also, we can apply these models to find suitable refugia in the past, for example, the last glacial maximum. I will provide here some example with Posidonia oceanica, which was the species I showed you at the beginning. It is a Mediterranean endemism, very important, and it has a temperate origin, and we perform a hindcast of its distribution to the last glacial maximum and compared also this with the genetic structure that other studies found. So these cells you can see here are the genetic regions found by these studies. And we found that in the past, the area with higher probability of occurrence was in agreement with the regions, the genetic regions, where higher allelic richness and private alleles were. So we found that the Cyclo-Tunisian Strait could be a suitable refugia in the past, where when in the last glacial maximum, the ice sheet was covering most of Europe and the north of the Mediterranean Sea. When applied these models to the future, we can predict by means of projections, scenarios of different RCPs. And we did that also, besides Posidonia oceanica, also with Thymodocea nodosa, which is also an important seagrass, but this is a tropical species. So for Thymodocea nodosa also, we have data about the genetic structure, and we found that these species, these tropical species, could find in the future suitable habitat in the north coast of Africa, while Thymodocea nodosa could suffer a lot of loss of habitat and could suffer a functional extinction in the future. So this kind of study was also applied for 3 Cystocereid species in collaboration with Roberto Bonomo, who did the genetic analysis of these species that are distributed in the Mediterranean and the Atlantic Sea. And he found this genetic structure, and when compared the change in habitat in the future, we found that many populations that show a great genetic diversity with many private alleles and rich gene pools could be lost in the future, this area in the southern limit of the distribution of the species. While in the northern distribution of the species in these populations, some habitat could be gained, but these populations harbor a much lower genetic diversity because they are more homogeneous, because they come from a founder effect. They migrated from the south after the last glacial maximum, and they expanded, and so they have a more homogeneous genetic structure. We have applied also hybrid models to try to refine these predictions, applying these mechanistic models to marine invasions. Marine invasions are very numerous in the sea because ballast water, for example, and in this case, Sargassum muticum, which is a seaweed that is a very good colonizer because their reproductive traits, and also it's a pseudoperennial species which has drifting tally that can attach elsewhere. It comes from Japan, and have expanded through the north coast of the Atlantic, in Europe, and also in North America, in the west coast. And we try to integrate the reproductive phenology of the species in the models, because we knew that these species have a narrower thermal niche for reproduction. And observing the results of many papers around the globe that had studied this reproductive window, we found that from 10 degrees to 27 degrees was the reproductive window for the species. We also wanted to integrate day length because it's a factor that also seems to affect the reproduction of the species, but we didn't find that this factor could be a limiting factor in the distributional latitudinal range. So, taking into account that reproductive window, we could filter that some regions that were considered as suitable by the correlative models could not be suitable for the reproduction of the species because it was out. All this region in blue means that it's out of the reproductive window. So Arctic areas could be not possible for the reproduction of the species and the installation of persistent populations. Other cause of the increment in marine invasions is tropicalization. Many species are pushed by currents or also entering by entries at the Suez Canal. And this is the case of Alophila stipulacia, which is a species native in the Red Sea and the Indian Ocean. And it came across the Suez Canal a century ago and has expanded through the Mediterranean Sea, through the Eastern Mediterranean Sea. This tropical species, Marlene Wesselmann did some mesocosm experiments to test their thermal limits, both of the native and invaded populations. And she found different fundamental niche ranges for the two populations. And we then integrated that in the models, providing a hybrid model of correlatives and thermal niches, both from using the thermal range, for the local populations, the native ones, and also for the invaded regions. And we found that mostly the differences in the models were found in the Mediterranean Sea, suggesting that obviously there was a local adaptation. Also, our models integrating local adaptation of these marine species was more accurate, while the one integrating the thermal niches of niche conservatism hypothesis had many wrong occurrences not predicted by the models. Okay. Another issue related to climate change in the oceans is the sea level rise. Sea level rise is affecting nowadays the coastal area shorelines, and it's also predicted to increase. As you can see by by 2100, depending on the scenario, it could suffer a rise of half meter to 80 centimeters. And the trend is also increasing when we see the historical data. So we wanted to combine the effects of gradual warming with the sea level rise in our predictions. And we wanted to study a region which has a very high primary production. In this map, you can see that the pink or red regions represent the highest primary production. And you can see here, like a point in the sea. This is the bank of Argan, which is in Mauritania. And there are seagrass meadows and a flat extension very wide. And so in this paper, we wanted to combine 3 dimensions. One was the time, because we wanted to predict the future, but taking into account not only the latitudinal shift that the species could suffer, but also taking into account the vertical niche of the species, because we wanted to include the depth, to estimate also the sea level rise and their effects. So we used 3 seagrass species, which are the only ones present there. They have different characteristics because Allodule variety is a tropical species and it is subtidal, as well as Thymodosea nodosa, but this Thymodosea nodosa is a temperate species, and Thostera nodotae, which is the only intertidal species there, and also a temperate species. So for this last species, the bank of Argan represents their southernmost distributional limit. So it was very interesting to test this hypothesis because also they play different functional roles in the ecosystem because they have different width in their leaves and also different shoot density in the meadows. So this work involves a lot of intense work with GIS because we had to create a bathymetric, combining bathymetric data and digital elevation models from land to form a digital terrain model combining land and sea. And that was difficult because there wasn't very accurate data for this region, not even LIDAR. And we finally obtained these bathymetric layers. This one in the left represents the subtidal ecosystems, and this one the intertidal. And we had to create these layers for every time moment according to the projected sea level rises changes regionally. So in the end, we obtained these predictions combining climate change and sea level rise, and we found that in the future, probably only Allodule wrightii, the tropical species, could be present there because most of the intertidal habitat could be lost. Also, we projected some changes in the sheet northwards that could contract the range of Thostera nodotae and Thymodosea nodosa, while Allodule wrightii could expand northwards. So these are some examples and showing that gradual warming is affecting these species, but we think that there are many other factors related to temperature, not talking about other factors such as anthropogenic factors or local factors, that could be related to persistent and extirpation of populations. So, for example, extreme events are increasing in intensity, frequency, and duration across the globe. They're not affecting equally all the regions, as you can see, but it could suffer a synergy. There could be a synergy between gradual warming and marine heat waves, for example. As you can see, they could increment. Also, it is predicted an increase in this trend for this kind of extreme events. This is impacting many ecosystems, not only macrophytes, but also animals in the marine realm. And there are also other factors that haven't been included in previous works studying macrophytes very much. And when I had the opportunity to collaborate with this team that is focused on phytoplankton and oceanography, and in this nice work led by Miriam Gerard, I could then realize that the temperature variability can be also affecting the distribution of species, and it is important to try to integrate these factors also in our predictions. So in the next research, I will show you, which is vinculated to my Marhot project, we are studying the factors that are affecting the extirpation and persistence of macrophytes population of macroalgae in the north of the Iberian Peninsula. These macroalgae are 6 cold temperate species which are distributed in the north of in Europe and also some in the America, but their ranges have been contracted northwards, following the common latitudinal shifts of other species. But also in the north of the peninsula, in this coast, there's suffering a contraction westwards, longitudinally. Since the 90s, it has been observed and registered by publications that they have been moving to the west. So you can see here some samples. This is Fucus, Serratus, I don't remember Serratus of vesiculosus now, and you can see how they moved in the time, and it was replaced by other kind of algae like ulva species, turf, which doesn't play the same role in the ecosystem because these are not structuring species, habitat-forming species. This pattern is common with other laminarias and other species such as Saccharissa and Imantalia. So we're studying why some populations persist and others are suffering extirpation. And to study that, we've been using time series of sea surface temperature, and we divided the series in a baseline and a second period. The baseline period, these years where the persistence of the populations were constated by surveys, and the resurvey period is this one. So we found that some of the variables, such as, for example, the mean sea surface temperature in autumn, could be affecting the extirpation, which is in black here, of these populations of all the 6 species, could be related to that. But to integrate other factors, besides the gradual warming, we also tested extreme events such as marine heat waves, and also we wanted to test marine cold spells, and also temperature variability with some variables not very used in this kind of works. We found up to now, because this is in preparation, that not all the species are affected by the same factors. Each species is related, its disappearance with a different factor related to temperature, but we found that there's a pattern when we take the sum of the variables, as these 3 that are shown, the sea surface temperature in autumn, and skewness and seasonality that are related to the temporal variability of temperature, then we see a pattern that discriminate between the persistent and the extirpated populations. This pattern is also found here in this NMDS, where you can see that the environmental envelope of the persistent and extirpation of the extirpated populations are very different and is very well separated. So we're still working on it to relate this with the life cycle of the species and to try to find a reason, a common pattern. I've been also in this Marhot project, in the Marie Curie project, trying to perform experiments to test the thermal limits of these species. So I was with Francisco Arenas. Sorry, I didn't name that. This is Rosa Viejo and Brezo Martinez from the University Rey Juan Carlos, and Francisco Arenas works in the CIMAR, which is in Matosinhos, Portugal. So I went there last year, and we performed an intensive mesocosm experiment to test the thermal limits of 2 species of macroalgae using 2 populations, 12 sea surface temperature treatments, and 2 air treatments, because these are intertidal species and are also affected by the temperature of the air. So we perform also PAM image to test the photosynthetic activity of the samples once they have applied the treatment. We study their growth, their respirometry, etc., and also carbon-nitrogen ratio. We're still analyzing this data, but we are trying to incorporate it in mechanistic models, integrating these data, these thermal limits, with correlative models. So how do we continue? Because when I went to the IBS meeting in Vancouver, I could be with many experts in marine chains, biogeography, and we found that there's a common pattern in many regions that we are observing here in Spain, in the north of the Peninsula Iberica, many patterns that has been found also in California, Canada, or Chile. And for example, a week ago, this paper was published, and they claim that there's a need to take some coast, rocky shore coast, sentinel coast, because there are many changes that should be monitored globally in many around the world. So this is our idea, to have a sentinel coast in the northern Iberian Peninsula. And along my research career, I've been integrating different techniques to study from a biogeographic perspective, using firstly correlative models, then mechanistic ones, using spatial analysis, also surveys. I couldn't show you this kind of research, physiological experiments. And now I will go to another step forward to integrate field experiments, because we have now 2 projects that were funded by the Ministerio de Ciencia, and will begin next year. We'll be working, it's a collaboration with my colleagues in the Universidad Rey Juan Carlos, and also José Luis Acuña and Julio Arrontes from the Universidad de Oviedo. We will do many field experiments in this coast, testing why persistent or extirpation or populations are occurring in different locations. And we will apply a integrative perspective, combining different scales. So, well, I would like to thank many of my co-authors that I couldn't mention here, but they're very important. And also, I couldn't show you some secondary lines I'm researching, and many other works that I've done with corals, with Pepe Templado, which is here, and others that I did in vegetation of saltworks of Ria Formosa, and vegetation of dune habitats with Sergio Chozas. And now I'm also collaborating with Moses, with people from the museum, with Manuel Molina and Joaquín Hortal. So, well, this is all. Thank you.
Thanks, Rosa. That was really, really interesting. So there are many quite shocking changes that you've shown that have happened very quickly. But for me, maybe the most shocking one are those changes you've shown in the local extinctions of those 6 species of brown algae. along the Cantabrian coast of Spain. And so you mentioned that in some places that those brown algae are being replaced by green algae like ulva. And so, I mean, in like 25 years, what does that mean for the structure of those algal communities and the functioning of those ecosystems and the animals that also depend on the macrophytes occurring there?
Yeah, you are right, because ulva species are cespitous species. But, for example, the macroalgae that were before, Fucus, Saccharisa, laminaria, are food for fishes, for example. In fact, we've found an increase in marks of fishes that are feeding in this type of macroalgae. So the functional role of these different communities is very marked. Also, there's a simplification, because you can see that there was only some species, but sometimes the rock is empty, totally empty. Not even ulva species or other cespitous species are there. Turf is also named.
And so that was, most of those disappearances have happened along the north coast there. On the Atlantic coast, down in Galicia and the north of Portugal, maybe, although from looking at your map, it looked like the sea had been warming as much or even more in those areas, but the species haven't been lost. Is that because the sort of the baseline temperatures were much lower there? And have you seen colonizations by those brown algae into any new locations as well?
Well, the coast of Galicia suffer from upwelling, so the water there is cooler, and they provide a much better habitat for these species up to now, because it is also discussed if there is awakening in this upwelling effect. We found some early signs that can imply that also. So up to now, it has been some kind of refuge for these species because of that upwelling of cooler water from the depth.
What you're essentially suggesting is that in the next 50 years, the macroalgal communities of the north and northwest coast of Spain are just going to be completely different.
It could be
what everyone is used to.
Yeah, maybe we can see a redistribution of species, and these species, this is the southernmost limit of most of them. So in northern populations, they are very healthy in England, and so maybe there would be a replacement of these species, many by marine species.
Yeah.
Invasive species, I mean.
Yeah.
Can I jump in? Because is this having any effect? I'm thinking about the sea, or some key fishing species there.
Yeah, for example, seagrass species have been found to be related with the buffering of temperature, for example, for clams, which are harvested in some areas in Galicia, for example. And fishes, of course, fishes.
Anchovies, for example, anchovies were...
Anchovies.
Anchovies. Were
the Cantabrian anchovy. I have no data right now about the Cantabrian anchovy, but because anchovy is it migrates a lot, so maybe it doesn't feed right here.
Okay. Thanks for the presentation. And did you test or evaluate or just see if some species going deeper side to avoid the climate change? Or
Yeah.
something?
Yeah, it is happening that some species are finding cooler water in the depth, some laminarias, for example. So they're disappearing for shallow waters, but you can find it if you dive in a deeper range. Yes.
Well, thank you for the presentation. I have another question with about the local extinctions. In the period that you analyzed from early 90s, I think, or 93 to actuality, there is the event of the prestige with a lot of black tide. Do you think that this event affect the local extinctions, or?
Well, in fact, the regions, the prestige was in this area, and there's still persistence. Maybe local extinctions exactly around this coast, probably in the short term, but maybe that populations could be recovered by now.
More questions? I've got one. I'm interested in those. You've shown here that there are some persistence populations here. You can see also in the NNDS that are within, more or less within the region where you've got most of the extirpations. Why is this? Why are those populations persisting? Do they have any? Yeah,
you mean, for example, these red?
Yeah, well, in these two cases, I mean, it seems that the change in temperature, this surface temperature is not that big. But in the ones that are a bit more,
yeah,
there, which are in the area of extirpation. I mean, it's a massive extirpation of everything, but still you've got a few populations. And if you see the NNDS, they are in a particular region of the climatic envelope. Do you think this is something? They are in that area. There is something there that allows some climatic conditions that are not that harsh, or is just particular populations of a?
As with the data we have in this study, we cannot test that because we're using a resolution of 0.25 degrees. Okay. So that's something that we are very interested in testing in the next 2 projects, because we want to do local experiments in the field to test why populations are suffering extirpation or persist at a micro scale. And also we want to take some restoration actions for these populations in places we think that it can be a persistence in the future.
Okay, you're going to target some of those populations.
We will target mostly populations that were historically but suffer extirpation, and also in populations where that still persists. We will contrast that.
Another question? Have you got anything from? Okay. So thank you, Rosa, for the nice talk. And if any one of you want to chat with her, you can remain here, or we are going to head to the residency after lunch.
Thank you. You're welcome.